Hey guys, Assalamu alaikum everyone. I hope you all doing well. This is the last part of our chapter biological molecules, and in the previous part we have done till here. We finished carbohydrates and we were on the structure of cellulose, right? Now in this part we are gonna start proteins. What are proteins? Proteins are basically complex biomolecules. Why are they known as complex? Because these are made up of many amino acids, and together they have the linkage that is known as the peptide linkage. Okay. Now these are made up of several elements, for example, carbon, nitrogen, sulfur, oxygen. Okay, and these can also be made up with phosphorus. What are proteins? The simplest definition is that these are polymers of amino acid. Now, what is a polymer? Polymer means when basic units like monomers they combine together with a linkage in between, for example, a peptide linkage, in order to form a polymer. Polymer is a complex structure, right? Now, what is the basic unit of proteins? Protein has got an amino group, carboxyl group, hydrogen atom, and an R group or the side chain. As you can see over here, this is the R group or the side chain. This is the amino group, which is NH2, right? This is the carboxyl group, okay? And this together, this OH of the carboxyl group and the H of the amine group, these two, they form what? They form water, okay? And when they form water, what happens over here is that it forms a peptide bond or the peptide linkage, okay? So in this bond, C double bond O, and then I have N and H. This forms a peptide linkage, okay? After that, we have CH3, and this is the whole structure, okay? Right? So we have a hydrogen atom, amino group, carboxyl group, R group, that is also known as the side chain, right? Now there are different properties. Proteins are amphoteric. Amphoteric means that they can act as an acid or a base, okay? And this property is known as the property of being a Zwitter ion, okay? When there's a possibility of the element being an acid, or it can also be a base. Now, these are classified as essential amino acids, which means that these are obtained from their diet. And non-essential amino acids are synthesized in our body. These are essential. Why? Because our body cannot produce them, and these must be obtained from what? From external sources. Okay. And the non-essential amino acids, these can easily be synthesized into our body. All right. Now see, the primary structure is a polypeptide chain, right? And the secondary structure is the alpha helix and the beta pleated sheet. Okay. These are produced by hydrogen bonds forming within the polypeptide. The first structure is simple. It is straight. As you can see over here, it is a linear sequence of amino acid, and the example is an insulin chain. Okay. The secondary structures are alpha helix or beta pleated sheet, which are formed when the linear sequences they combine together with the help of the hydrogen bonds. Now, what is tertiary? Tertiary is the 3D folding of a single chain, and the example over here is myoglobin. And quaternary is multiple chain forming complex, and the example is hemoglobin. See over here, tertiary structure is the random coiling structure over here, and this is the region of the secondary structure of the alpha helix, right? And it is a 3D overall folding of the protein containing the secondary structures. This is the tertiary structure. And what is quaternary? Quaternary means that two or more polypeptides, these are jumbled up together into a 3D like structure, and the example over here is hemoglobin, whereas for tertiary it is myoglobin, right? Now, what is the significance of amino acids? As we all know, it is the building block of proteins, provide energy source. Okay. Now, what is the precursor? It is a precursor of important biomolecules. What do you mean by precursor? It means that it is a foundation stone for the development of many biomolecules. For example, the development of several hormones, neurotransmitters, heme, purine, and pyrimidines, right? It is also used for the nitrogen balance, which is essential for growth and repair, and as enzymes. Okay, so all the enzymes, hormones, neurotransmitters, pumps, pyrimidines, right? All of these are made up of amino acids. Now, what is the significance of proteins? You guys can, take a screenshot and read this, because this is like, very easy. It provides a structural role. For example, collagen, keratin, and elastin. These give the strength to hair, nails, skin, and tendons. It provides the function of enzymes. It helps in transport. For example, hemoglobin, it is present in the RBCs, red blood cells, and it helps in the transport of oxygen. Next, we have got plasma proteins in the form of albumin and membrane transporters. For defense, we have got antibodies, right? which is the immunoglobulins. Okay, so these help in defense, these help in movement, storage. Why? Because ferritin is used for iron storage, and casein is a milk protein that is used for storage as well. Next, we have buffering capacity, osmotic balance. Why? Because plasma proteins, for example albumin, they maintain the oncotic pressure. What is oncotic? It means basically the pressure of proteins. Okay, and it provides energy source. Proteins provide the energy of 4 kilocalorie per gram when carbohydrates and fats are insufficient in our body. Now, these are the structural proteins. This is the table which I took from the Federal Board Biology, right? So, the first one I have over here is collagen, is a structural protein, right? It is made up of basically it establishes a matrix of bones and cartilage, and ligaments are also present over there, ligaments and tendons, so it provides structure to our body. Next, we have got elastin. Elastin is present in connective tissues. Please remember this. This is important for your MCQ point of view, that it is present in tissues and it helps in tendons and ligaments. Next, we have keratin that strengthens the protective coverings, for example, hair, nails, quills, feathers. It is present in beaks. Next, we have histone that arranges the DNA inside the chromosome. Okay. So these are the specific functions. You need to remember them for your MCQ point of view. Now we move on to lipids. What are lipids? Lipids are heterogeneous group of organic compounds. Okay. These are insoluble in water, but these are soluble in non-polar solvents. For example, ether, chloroform, and benzene. These are important for energy storage, structure, and signaling. Okay, these are soluble in non-polar solvents. Why? And why are these insoluble in water? Because water is polar. Okay, it breaks down into H positive and OH negative, right? It is polar, it is hydrophilic. Whereas lipids, these are hydrophobic, right? When these are hydrophobic, it means that they move away from water. These are insoluble in water, but they are soluble in organic solvents. For example, we have ether, we have got chloroform, and we also have benzene. These are used for energy storage, structure, and signaling. So lipids provide basically 9 kilocalories of energy. Carbohydrates and proteins, they provide a lesser amount of energy. Okay. Now, what is a phospholipid? Phospholipid is a glycerol backbone along with two fatty acids, a phosphate group that is linked to choline, ethanolamine, and serine. Okay, the nature is amphipathic. It has got a hydrophilic head and hydrophobic tail. As you see over here, this head is the hydrophilic head. Why is it hydrophilic? Because it contains phosphate backbone, and it also contains glycerol. And we have a tail that is hydrophobic, sorry. Why? Because it is made up of fatty acids, and fatty acid is a lipid, right? So lipid is hydrophobic, right? The first one we have over here is saturated fatty acid, that is basically made up of the single bond, and it is structure just like of an alkane. But over here we have unsaturated fatty acid, and this has a double bond, right? And double bond means a structure like an alkene. Alkene has got double bond in between its carbon atoms, right? So why is unsaturated fatty acid like this? Why? There's a reason that it causes the kinking of the unsaturated fatty acid chain. Why? Due to the presence of the double bond carbon atoms. Okay. Now, the functions of phospholipids is that it is the main structural component of the cell membranes. It is a component of the phospholipid bilayer. It is involved in signaling, and it is also involved in the fluidity of the membranes. The example is phosphatidylcholine and phosphatidylserine. Okay, so these are the examples of it, and it is involved in the cell signaling, and it is also involved in fluidity, right? These are a part of the cell membrane. Next, we move on to the triglycerides. What is a triglyceride? These are the neutral fats. These are made up of glycerol plus 3 fatty acids via linkage that is known as the ester linkage. See over here, this is glycerol, and these are the 3 fatty acids. Now, this linkage which helps to join the glycerol and the fatty acid, this is known as the ester linkage. Okay, what happens over here is that basically water is formed. Hydrogen is removed and one hydroxyl is removed, and it forms water. Okay, so when water is formed, the linkage that is formed is known as the ester linkage. The nature is hydrophobic. It is stored inside the adipose tissue. Okay, it is stored inside the cells which help to store fat. Okay, that is known as adipose. Basically, the literal meaning of adipose is fat. It is an energy reserve. It stores 9 kilocalories per gram, as I told you before. It provides insulation, protection, and it is stored for long-term energy needs. Okay. Now we move on to esters. What is an ester? It is formed by the condensation of fatty acid with glycerol. As you see over here, see, this is glycerol and this is the fatty acid which has a R group, and this is the condensation that forms a linkage that is known as the ester linkage. Okay. So what happens? Monoacylglycerol, it means one fatty acid plus glycerol. Diacylglycerol, two fatty acid plus glycerol. Triacylglycerol, three fatty acid per glycerol. Okay. So mono, di, tri, tetra for four, penta for five, hexa for six, hepta for seven, octa for eight, nana for nine, and deca for 10. So just like we studied all the shapes when we were a child, like triangle, then we studied about the pentagon, hexagon, heptagon, octagon, nonagon, all of these, right? So similarly over here, as I keep on increasing the numbers in Roman or Latin or whatever language is this, what I do is that I keep on increasing the fatty acid as well, number of fatty acid as well. This is a triacylglycerol. Why? Because there are three molecules of the fatty acid over here, right? Okay, and there is one glycerol. Okay, so this is steric, this is palmitic, and this is lauric. It depends upon the R group. Okay, this is stearic acid, palmitic acid, and lauric acid, right? Now we move on to DNA. We are on nucleotides, which is our last topic now. Sorry, not last. We have got conjugated molecules as well. So we have got DNA. DNA is the genetic material in most of the organisms. It consists of antiparallel polynucleotide chains. It forms a double helix-like structure like this, somewhat like this. My drawing is very bad. And it stores and transmits and expresses genetic information. Now there are different kinds of nitrogenous bases. The first one which we are going to study are purines. These are adenine and guanine. And the second one is pyrimidines, which are ringed. Now see, look at the structure over here. These are purines. Purines have got two rings. Okay. One and two. One and two. And what are pyrimidines? Pyrimidine has got a single ring only. Okay. So purines are adenine and guanine, and pyrimidines are uracil, thymine, and cytosine. Now uracil is present where? It is present only in RNA. Thymine is present only in DNA. Cytosine is present in both DNA and RNA. Okay. So whenever we have got DNA, we're going to write thymine, and whenever we have RNA, we're going to write uracil. And adenine and guanine and cytosine, these three are present in both, whether it is DNA or RNA. You just need to remember that adenine and guanine are purines, which means that they have got double ring structure, whereas pyrimidines, which are cytosine, thymine, and uracil, they have got a single ring structure. Now we move on to gene. What is a gene? Gene is a specific sequence of nucleotides which are present in the DNA. As you see over here, these are the different set of nucleotides which are present in the DNA. And this is a gene. Okay, this is a specific structure. It carries a genetic code for the synthesis of the polypeptide or the functional RNA. So it carries the genetic code. As you see over here, these three bases, nitrogenous bases, they form a codon. Okay, this codon is a specific message that helps to, whenever all of these codons join together, what they form, they have to form a protein, or they form a gene for a specific characteristic which is present inside the human body. Each gene has got a promoter region that helps to start the transcription. Next, we have the coding region or the exons. They contain instructions for the amino acid sequence, and the non-coding regions, which are known as introns, they control the gene expression. Okay, so there are basically promoter region which helps to start, it helps to initiate the transcription. Next, we have the region that performs the coding, it is the coding region. Okay, and these contain instruction for the amino acid sequence. Next, we have non-coding regions that are known as the introns. Okay, so this is how the protein chain is formed. The first one is the promoter region, then we have the coding region, okay, exons, and then we have introns as well, which are the non-coding regions, right? So in this way, a protein chain is formed. Okay, now we move on to the double helical structure of DNA, that is the Watson and the Crick model. Now, what they said is that the DNA is a double helix structure. We all know that two polynucleotide chains are twisted around each other, whereas RNA is a single-stranded structure, DNA is a double-stranded structure. Now, these two strands run anti-parallel. One is in 5 to 3, okay, and the other one is 3 to 5. Okay, so you don't need to go into the depths of this. You just need to see that it is basically on the basis of position, and it is that both of them are like this, going anti-parallel. Okay, some if I look at the road and these are two cars, these are coming towards you like this. Okay, so these are parallel. But if these two cars, one of the car is coming like this and the other car is coming like this, so these two are going anti-parallel. Okay, one of the car is coming towards you and one of them is going away from you. Similarly, one of the gene, for example, this strand starts from here and this strand, it starts from here. Both of them are making like this. So one of them starts from 3 to 5, that is the leading strand, and the other one is the lagging strand, which you're going to study later. Held together by the hydrogen bonds in between bases. What are present between the bases? These are hydrogen bonds. Now adenine pairs with thymine and guanine pairs with cytosine. Adenine pairs with thymine. This is complementary base pairing. Okay, the shape of adenine and the shape of thymine, these are complementary and they fit together with the help of two hydrogens. And cytosine and guanine, these fit together with the help of three hydrogens, and this ensures complementary strands. Now we have got a backbone. Each strand has got a sugar and phosphate backbone, that is deoxyribose plus phosphate on the outside. See, this is a sugar and a phosphate backbone. Okay, and these are the nitrogenous base pairs on the inside, whereas the sugar phosphate backbone is present on the outside, and these are held together by the hydrogen bond. So this is the Watson and the Crick model. One full turn is equals to 34 angstrom. Angstrom is what? Angstrom is the unit of wavelength. You must have studied this in physics. If not, just remember this for now. The bases inside are nitrogenous bases. They face inwards and they're stacked like the steps of a ladder. You see that they form a shape just like we have a ladder, right? They form a shape like that. Okay. So the distance between the adjacent bases is 3.4 angstrom. Okay. Whenever we have the adjacent bases, the distance is 3.4 angstrom, and the distance between one full turn, it is 34 angstrom. Okay, the diameter of the helix is 20 angstrom. So you see, if I have measured this distance, it is 20 angstrom. Please remember all of these distances. These are very important. Their stability is because of hydrogen bonding, and they have base stacking and interactions which help to stabilize this helix-like structure. And the importance is that it ensures accurate replication. Each strand acts like a template. Okay, so what happens is that adenine has to pair with thymine. It cannot pair with any other molecule, whereas cytosine has to pair with guanine. It cannot pair up with any other molecule, and it stores genetic information inside the base sequences. Okay, RNA. RNA is a single-stranded nucleic acid. It is made up of ribonucleotides, ribose sugar plus phosphate plus a nitrogenous base, this adenine, uracil, guanine, and cytosine. Its function is that it acts between the DNA and protein. Okay, so what it does is that it helps in transcription and translation, and it helps in the formation of protein, which is the product. Okay, now we are going to study the types of RNA. Wait. Okay. So, what is messenger RNA? It basically carries the genetic code from the DNA to the ribosomes, that is the cytoplasm. It contains three codes that specify the amino acids, and it is synthesized during transcription. So, I'm going to show this to you. See, this is the process of transcription. What happens is that this is a double-stranded DNA, and by the process of transcription, it breaks down, it makes complementary strands, that is known as the single-stranded RNA. So it is used in the formation of proteins. It acts as a template in protein synthesis. This is very, very important for your MCQ, and it carries the genetic code from the DNA to the ribosomes. Okay. What it does is that it helps to transfer the code. And what is transfer RNA? It is a small RNA molecule that carries specific amino acids to the ribosome. It does not help in the transfer of genes. It does not act as a template of protein synthesis, but it helps in the transfer of specific amino acid. It has got an anticodon region which is complementary to the messenger RNA codon, and it matches the amino acids with the codon during translation. The role is the adapter molecule between messenger RNA and amino acid sequence. Okay, it acts like an adapter molecule between the messenger RNA and the amino acid sequence. Let me just show you. Wait a second. Wait a second. Just see. This is the structure of the transfer RNA. See. Okay, so this is the amino acid attachment site, right? It helps to attach amino acids over here, and it has got an anticodon loop over here as well, right? Wait, over here on the bottom. Okay, right? So you just remember that it helps to carry specific amino acids. Okay, now we move on to the ribosomal RNA. It is a structural and functional component of ribosomes. It combines with proteins to form small and large ribosomal units. It catalyzes the peptide bond formation and provides a site for translation. So in transcription, what helps? Messenger RNA helps. And in translation, what helps? Ribosomal RNA helps. Why? Because it helps in the formation of the peptide bonds. It is a component of ribosomes. It is present inside the ribosomes, and it helps to form the peptide bond formation with the help of peptidyl transferase. Okay. Now, this is just a summary that messenger RNA, in the location of messenger RNA, very important. It is present in the nucleus and then it goes to the cytoplasm as well. It carries the genetic code to the ribosome. Okay, and then we have transfer RNA that is present in the cytoplasm. It brings amino acids to the ribosome and it matches the codons. Ribosomal RNA, it forms the ribosome structure. It catalyzes the peptide bonds. It helps in the formation of the peptide bonds and it helps in translation. Messenger RNA helps in transcription, right? Ribosomal RNA helps in translation. Small nuclear RNA, it is present in the nucleus. It helps in RNA splicing. This is not important right now. We're going to study this later in the later chapters. And we have these two as well, which help in gene regulation, okay? Which are not that important. You can just remember or take a screenshot of this table, okay? So now we have got the process of transcription and translation. After that, we have got conjugated molecules. So what happens in transcription is that in the nucleus, the cell's machinery, it copies the gene sequence into the messenger RNA. What happens is that this DNA, double-stranded DNA, it breaks down into single-stranded RNA, and this RNA is known as the messenger RNA. Messenger RNA contains the blueprint that is used for protein synthesis later, okay? It is a molecule that is similar to DNA. Like DNA, messenger RNA has got 4 nucleotide bases, but in messenger RNA, the base uracil, it replaces thymine. This is very important that since it is an RNA and an RNA, thymine cannot exist. We have got what? We have got uracil. So adenine will be formed as a complementary for uracil instead of adenine forming for thymine in RNA, right? So we have got a single-stranded messenger RNA. Now this messenger RNA goes from the cell's nucleus. It travels into where? It travels into the cytoplasm, and in the cytoplasm, what happens? Translation happens. For translation, we need the ribosomal RNA, right? What happens over here? The protein-making machinery that is known as the ribosome, it reads the messenger RNA sequence. It goes from the nucleus to the cytoplasm to the ribosome specifically, and the ribosome reads the sequence. It translates it into the amino acid sequence of the protein. What it does is that it makes a complementary strand for an amino acid, right? Now the ribosome starts at the sequence AUG, then reads 3 nucleotides at one time, okay? So it starts at the sequence AUG, which is the start codon, okay? And after that, it keeps on forming more and more codons that help in the formation of protein. It specifies a particular amino acid. As we saw over here, see, It starts where? It starts with AUG, and after that it forms different proteins, okay, different amino acids which are combined in order to form proteins, okay. So this is how the amino acid chain is formed, right? And it is formed with the help of ribosomal RNA. The stop codons are these UAA, UAG, UGA. Please remember these. Start codon is AUG and the stop codons are these three UAA, UAG and UGA. They tell the ribosome that the protein is complete or not. Okay, so this was the whole proteins as well. If you have any queries, let me know. Now we move on to conjugated molecules or conjugated lipids. Okay. Now, what are conjugated lipids? They contain a lipid component plus another non-lipid component, that is a protein, carbohydrate, or a nucleic acid. They play specialized structural and functional roles. Okay. So, what happens? Conjugated means combined. Okay. Whenever a lipid is combined with another non-lipid. Conjugated lipid means that the major component will be lipid, but it also adds another food group, that is protein, carbohydrate, or a nucleic acid, right? The first one is glycolipid. Okay, lipid plus a carbohydrate. It is found in the cell membranes in the outer leaflet of the cell membranes. Glycolipid. Glyco means glucose or glycogen, right? And lipid means lipid. So it is lipid plus carbohydrate simply. And where is it found? It is found in the cell surface membrane. The function of it is cell recognition and cell communication, receptor sites for toxins and viruses, and examples are cerebrocytes and gangliocytes, which are present in the nerve tissue. Okay, these are the present in the cell membrane of the nerve tissue, and these are cerebrocytes and gangliocytes. You need to remember these. Cerebro from what? From the cerebral hemisphere, and ganglio from what? From the ganglions. Ganglions are present in the nervous system. Okay, so this is how you can remember this. Next, we have got lipoproteins. Okay, what is lipoproteins? Lipo means lipid and protein means protein. So simply lipoproteins, it transports lipids, for example, cholesterols and triglycerides in the blood, and its types are chylomicrons, very low density lipoprotein, low density and high density lipoproteins. So you need to remember the types, you need to remember the function, and you need to remember the function, and it helps in lipid transport and it also helps in lipid metabolism. So it is used to move the lipid from one molecule to another. Glycolipid was helped to make the cell membrane, okay, especially in the nervous system, and the examples were cerebrocytes and gangliocytes. Whereas lipoproteins, these are used for the transport of lipids, for the transport of very low density lipoprotein, low density and high density lipoproteins, right? And these are used for the lipid transport and metabolism. Now we have got phosphoproteins and nucleoproteins. Phosphoproteins, which means phosphate plus proteins, okay? Nucleoproteins mean nucleic acid plus protein, okay? Lipid plus protein plus phosphate or nucleic acid. Example is the plasma membranes when proteins are conjugated with phospholipids. So proteins are present with the phospholipids and these are found in plasma membranes. These are also found in, these are found in plasma membranes. Now we have nucleoproteins, which means nucleic acid plus proteins. These are found in the chromosomes, which are present in DNA or the DNA and proteins, right? Now we have got chromolipids. Chrome means what? Color. So it means lipid plus pigment. The examples are carotenoids and chlorophyll. Chlorophyll is green, carotenoids is orange shaped, orange like, right? Orange or reddish, right? Function is photosynthesis. These provide color in plants and animals. The significance is that it provides structural stability. It helps in transport. So this is the significance of all of the conjugated compounds, right? Conjugated products. So this is just a summary of the whole topic which we just studied, that it helps in transport. Which one helps in transport? It is basically the lipoprotein. Structural stability, it is basically the glycolipids, which is a form of the cell membrane. Then we have signaling, recognition and defense mechanisms. It helps in nervous system function. For example, myelin sheath, they contain glycolipids as well. Okay, so these are some of the MCQ points which you need to remember. What I want you to do is that take a screenshot of all of these, slides, or I have the PDF as well. There's a number given below in the description box. Please click on that and you can easily get the slides. And I'm going to add you to a group that is focused on MDCAT 2025 preparation so that we can work together, inshallah. All right. Till then, take care. And we have successfully finished the whole chapter of biological molecules. I have also taught acellular life. Okay. And if you have any queries in this chapter, please let me know. And also, I have taught bioenergetics on my YouTube. So if you have any queries, let me know. And I'm going to share the whole playlist for biology. Till then, take care. Allah Hafiz.